Hello everybody. Today I'm gonna talk about DNA replication. DNA replication consists of two types of replication, which are conservative replication, which is unable to change, and the other one is semi-conservative replication, which is partially to change. Paired bases of DNA molecule described as a complementary. DNA replication begins at a specific point in the DNA molecule called the origin of replication site, which is here in the replication fork, which called this yellow area the helicase. The helicase is an enzyme which unwinds and separates a portion of the DNA molecule, and single-strand proteins stabilize the standards. The enzyme complex DNA polymerase, which is located here, uh, it engages the separated portion of the molecule. As you know, DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides, which the nucleotides are these enzymes which binds to each other. Therefore, replication begins as an enzyme called primase assemble an RNA primer. RNA primer is at the origin of the replication site. It consists of a short sequence of RNA nucleotides, complementary to a small and initial section of the DNA strand begins prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer, and thus begin the process of constructing a new complementary strand of the DNA molecule. As you see here, this one is the leading strand, which consists of the old strand, this yellow line, and this green line, which also called a new strand. It's formed after the old. It's formed when the helicase, because of the helicase, when it uh, like separated from the helicase, which is the replication fork. The RNA primer enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. One strand replicated continuously separating portion of original DNA molecule, while the other strand is replicated discontinuously in opposite direction, which is this is the direction of the new strand of a leading strand, and this is the direction of the unwinding of the lagging strand. However, while the other strand is replicated discontinuously in opposite direction, the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer, which the RNA primer are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into complementary strands.